Okay, thank you. And yeah, I want to present to you some of the problems or some of the things a developer should consider when developing a gene therapy or cell gene therapy protocol, and yeah, especially from the view of a manufacturer, which we are. Since 2 weeks, we are called BioNTech Innovative Manufacturing Services. We were OIFETs up to then, but we are still the same people and still doing the same stuff that we did before, which is mainly being a contract manufacturing or development lab for cell and gene therapy, and we do retroviral vectors, in vitro transcribed RNA, and also cell products, and that's what I'm talking about today. So basically, this is about a project that you want to bring into clinical trials, and you could call it, you have a CDXY CAR T cells for the therapy of melanoma. And obviously, the scheme is known to everybody in the room. So you have a patient, you take cells from the patient, you enrich for T cells, or you don't, we talk about this. Then you stimulate them and do gene transfer with a retroviral vector or gamma or a lentiviral vector or a transposon, and bring in the CAR of your choice. Then you expand these cells ex vivo for, there are multiple possibilities in terms of cytokines that you use, in terms of length of process that you use, in terms of the equipment you use for this. But then in the end, you harvest your cells, you do the quality control. Mostly you freeze your cells to get the time to do your quality control as is, and finally you certify the cells, and they are shipped back to the clinics and infused to the patient. So when you have, at some point you come to the conclusion that you have your CAR architecture clarified, you know which co-stimulating domains you want to have in your CAR, you have your SCFV, everything looks fine, you have it cloned into a retroviral vector or a lentiviral vector. The transduction protocol is established, you get 30, 50, 60% transduction efficiency. You have very convincing in vitro experiments, your CAR T cells, they kill the target cells as expected. You even have in vivo experiments in an animal model that show encouraging results. And the risk assessment shows you that you have a safe strategy, and you think your T cell manufacturing process is established. But if you look deeper, this might not be the case. Not everything is ready. So in terms of vectors, so in T-cell gene therapy, still gamma retroviral vector are used a lot, and a lot of preclinical experiments are done with the classical architecture of these vectors, LTR-driven. But it might be better to switch to a SYN vector for clinical translation. This is, of course, true because regulators like the concept of SYN more, but that has a reason, because obviously it is a safer concept, and so you definitely should think about should we go with our LTR vector into the clinical trials, or should we switch to SYN? Then you think your transduction protocol is established, but is it at the right scale? Can you really manage the target cell doses that your clinical protocol would need? And do you have GMP-compatible raw materials in your process? This might not be the case, but you will need this in clinical translation. The QC, the quality control tests, have to be designed and established. And as we had heard earlier from the Paul Ehrlich Institute, it makes a lot of sense to go for scientific advice meeting early on to Paul Ehrlich or to your regulatory authority and discuss this concept with them. Then that is even more important in the preclinical experiments. You might have very convincing data in your preclinical experiments, but have they been done with the the right vector, with your final vector in the final architecture, have they been done with a cell source that is relevant in the clinical trial? And so how comprehensive is this data package? This has to be discussed, and yeah, I just recommend you talk with the regulators if they agree to you, and also with the in vivo experiments. Are they done with the right vector? Are they done in a relevant model? We know, especially with CAR T-cell therapies, there might not be a relevant model, but still you have to address this and talk to this. But in the rest of my talk, I will concentrate on this part, and here go more into details and present the questions that are there and how they can be addressed. Okay, this is a typical transduction expansion protocol for a T-cell therapy. So it all starts with the harvesting of the cell, which is normally done by leukapheresis. It can also be done by peripheral blood draw if less cells are needed, but normally it would start with leukapheresis. And then the cells are stimulated, either the whole cells or the T cells. And then viral transduction takes part, cells are expanded, and cells are harvested and cryopreserved. But let's start here on this day. The leukapheresis has been done. You have two possibilities here. You can take the leukapheresis to your lab and start manufacturing. This is, yeah. Or you can freeze your cells. We will discuss the differences here. Then you can use the whole leukapheresis, or you can enrich for T cells, or you can enrich for subgroups of T cells. And yeah, so which cells are it that you want to have? So here are some considerations regarding this. So the question, do you want fresh or frozen apheresis? The advantages of fresh are definitely you have less manufacturing steps, so it's a more straightforward protocol. You will always lose cells when you freeze, so it is and especially with patient material that might be limiting the number of cells. And in some protocols, if you purify cell, they are easier to purify, especially when you look at Clinimax CD4, CD8 stimulation purification. It's easier from a fresh cell population. The frozen also has advantages, and the main advantage is a practical one, a logistical one, because obviously if you use fresh material, you are dependent on the whole process before, on the patient being available for leukapheresis, on the leukapheresis center, and then your manufacturing unit has to be ready to start any day with the transduction process. Whereas if you can discouple this and freeze the cells in between, you can start the transduction protocol when the clean rooms are ready for this, when the people are ready for this. And so from a practical view, I encourage everybody to go that way if there is enough cells, if cells is not limiting. Then about the enrichment of target cells. So the easiest you can do, or the lowest degree of enrichment you can do, is by doing a Ficol gradient enrichment of mononuclear cells. If you do this, I would recommend to do this in a closed system. There is equipment available that does this in a closed system. There might be a bit higher cell losses, but it's just a much more safer procedure. Then, of course, you can do CD4 and CD8, or only CD4 or only CD8 enrichment by Clinimax. This you can only do before freezing, because if you thaw the cells, they are clumpy and you have much lower recovery rates. So if you go for enrichment and freezing, on the first day you have these two steps to perform. And then also you can, of course, enrich your cells with dynabeads, the CD3, CD28 beads, 28 beads. This can be done after thawing, and obviously it's also a stimulating reagent that if you do CAR T-cell therapy is available to you, because it is it is not exclusively claimed or the IP is available, but if you do TCR therapy, this reagent is not available to you because there is an exclusive, or the IP belongs exclusively to one company, and so if you're not working with that company, you won't have this reagent available to you, and you have to, we come to that in a later slide. Okay, so you have decided on one of these ways. As I said, I would recommend you go for frozen products which might be enriched for CD4, CD8 cells. Then you have to stimulate them, and you need for that clinical grade stimulation reagents. You have to need cytokines, but which cytokines are the right ones? You obviously have to have the cytokines in clinical grade or in GMP quality. You will need clinical grade medium, but there are a lot of mediums out there that are suitable for T cells. You have to decide for one. And then most of the lab scale protocols using AB serum, even if the media claim to be serum free, most people still use AB serum in their culture. But yeah, you have to decide if you want to do this. Again, in the next slide, I have some consideration for that. So regarding stimulation reagents, I just told you that the dynabeats are not available to everybody in the TCR field. In the CAR field, they are available. There are other reagents that are available because you want to do CD3, CD28 stimulation. It's definitely the way to go forward. But you have to define what kind of cells. What I can say, you get different cells from these two reagents, and there are other reagents coming up now, and you have to decide if you are happy with what you get. You have to test them. Then, in terms of cytokines, there is most clinical experience is with IL-2. So most of the early days clinical trials have been done with IL-2 stimulation of T cells. IL-7, IL-15 is coming up, and it looks like the phenotype you get is more preferable if you use IL-7, IL-15. But again, the clinical experience is much more limited with these cytokines. The good news is that there are enough, or that there are multiple vendors that provide these cytokines in GMP grade, so that's not a problem. Then with the human AB serum, so it works, so that's very good, but it's expensive, it's undefined, there are lot-to-lot variation. It is regulatory difficult, especially if you work in the US and in Europe, because there are different regulations for testing the donors for this serum in Europe and America. So it's not easy to import US serum for German trial or for European trials and vice versa. And that's, I think, the most serious thing. There's a limited supply, and if CAR T cell therapies, they are designed to, if they are successful, there are thousands and thousands of patients that can be treated, and I doubt there is enough serum out there to do this. So there are alternatives emerging, but they need to be tested. So there are serum replacements available in clinical grade, but you have to test them in your protocol. And again, the cells that come out of these protocols, they are the cells that you have to test in your preclinical experiments and in your animal models. These are the cells, and it might be, if you have tested other cells, they might be different. Okay, we're coming to the viral transduction. That's typically done if you're using a retroviral, gamma retroviral vector on day 3 or 4 of the process, if you use a lentiviral vector earlier, because they don't need cell division. So with that, you need, or most people use a co-localization reagents, mostly retronactine. You have to have clinical grade of this reagent, otherwise, it's not possible. And then you have to decide, do you have 1 or 2 transduction cycles? Again, some consideration to this. So the clinical grade retronactine is available, so that's good, but it comes at a cost. So in our experience, retroenactine is the single most expensive reagent going into a patient. So the cost per patient are 5,000 to 10,000 just for this reagent. And then obviously the sponsor of the trial, the developer, also needs a license from Takara for this. I have no idea what they want, but I guess it's also some considerable amount. So it makes sense to think about alternatives, and there are some alternatives emerging. Miltony has brought out one reagent. This one is coming up, but not yet clinically available. But there is some movement on that front, and if you are not already there and want to start your clinical trial tomorrow, it might make sense to look for an alternative because it will influence your cost of goods in the end dramatically. Then about the transduction cycle. So obviously one transduction cycle is less expensive. You use only half of the vector bed. You have a lower copy number per cell, which for safety reasons is good. You have less handling steps, and you get more transduced cells per infectious particles, because normally, a typical transduction would look like that you have, let's say, 30% after the first transduction step and maybe 35 to 40 after the second with the same MOI. So you get more infectious, you get more transduction per infectious particle with one. On the other hand, if you use just one, if you use 2 transduction cycles, you get a higher transduction efficiency, which is obviously beneficial for your protocol. And especially with gamma retroviral vectors, you need proliferation for the transduction, and maybe if you just give one cycle, you might miss the right day. So you're much safer with 2 transduction cycles. You just have to decide what to do. After the transduction, your cells are expanded, and for numerous days it goes anything between 3 and 10 days expansion after transduction are typical, and there are different systems available for this, and they are discussed here. So what's not shown here is the most simple system would be just bags that cells can be cultured in, and they work fine, but they only work until a given cell density. So there are other systems that give you more cells with less handling steps. One of them are the wave bioreactors. They allow to adjust and control multiple parameters. So they are a bioreactor, and you can really control what's going on. You know what the pH is, you know what the O2 saturation of your medium is. You get a lot of insight on what's going on in your process, and you can generate the highest cell number with this system. But the establishment can be rather cumbersome because there are so many variables you can look at, and it doesn't work the same with all cells. So we have experience with cells that have a different purification scheme than others, and they behave totally different in the wave bioreactor than others. So you can't just say, this is my wave protocol and it works for every cell. It doesn't. So then there is the GREX system. It's rather the contrary to the wave in that it is not transparent at all. It's kind of a black box. You put cells in there, put them in the incubator, leave them there for a week, take them out, and there is not much, or there is nothing you can influence there. But the cells that come out there, they are nice. So it's even with these limitations, it's a nice system, and it's really worth having a look at it. And it gives very good cell numbers, very high proliferation, and a very good phenotype, and very low handling steps. It's very easy to establish compared to this one. And then there's the Prodigy system. The Prodigy offers the highest degree of automation and also of closedness. It's a really, you can really design a closed process with the Prodigy, but it comes at other expenses. It's an expensive machine, obviously, And there is much lower flexibility in terms of cell numbers, and we will come to cell numbers later, especially in phase 1 trials. Normally you want to span a very high number, or high degree of cell numbers. So, but yeah, very early on you have to commit to one of these systems and say, okay, this is the one I want to go with. So, and then the last topic is how long do you want to expand? When do you want to harvest? How do you want to harvest? And so this is largely depending on how many cells you want, how the cell shall look. The only thing that I say how they shall look. Obviously, you want to know how they function, but you will only know how they function later in the patient. So you look at them to kind of predict function. And then which harvesting method or device do you use? You want to be closed. Obviously, if you are in the Prodigy, then it's within the system. But if you're in Xuri or in the G-Rex, you need to have some other devices to harvest the cells. So some consideration to this. So one thing that's very important but very often overlooked is the process development is being done with healthy donor material normally, which might be totally different in terms of proliferative capacity and as well as number available for starting than patient cells. So when you talk about the cell numbers you want and you want to establish, always have in mind that it might be totally different in the patients, in your patient material than in the normal donor material that you used for establishment. Then, as I said already, phase 1 trials normally have a dose escalation scheme, and these goals, typically there are log numbers of cells. So typical, maybe from 10 to the 6th to 10 to the 9th per kilogram patient weight. So that's 3 logs. But they all have to be done with the same process. So, but obviously the process that you have established has to allow for the largest amount of cell, for the highest amount of cell number. That's the scale you establish because it can't be all the cells that you compare have to come from the same process, of course, and that can lead to longer than optimal expansion times, because obviously. From a cost perspective, but also from a cell functionality perspective, you want young cells. You want maybe harvest on day 7 or on day 10 of the process, but because you committed to cell numbers very high and committed to a cell expansion system, you find out during the process development that you have to culture for 14 days to reach these cell numbers. And maybe that's not beneficial because younger cells might be more positive. You can look at it, and we have looked at it in processes that we have established. So it looks like from a phenotype, you even get very good-looking cells, central memory and very early memory cells on day 14 of a process, but the functionality will only be looked at in the patient. And but then you also have to consider that the process has to fit for the lowest cell numbers. So your freezing protocol, your harvesting protocol also has to fit for this, and this might play a role in some of your QC assays, the flow cytometry assays, for instance, where you have to take a sample from your final product, and your final product is just 10 to the fifth or 100,000 cells per milliliter, whereas in the higher cell doses it's 1,000 times higher. So all this you have to consider when you design the process, and it makes sense to take your time with this, because once you have locked down your process and you have treated your first patients with this process, and then you find out, okay, it might have been better to be shorter, it's a lot harder to go back to, because you have to show comparability of the product you produced with your first process to the product you produce with your next generation process. So you want to have a process that is fit for the future, and that you don't have to change after each clinical phase. And that's basically my last words here. And I thank you for your attention, and I'm happy to take questions.